Hi everyone. In this video, I'm going to be talking about testing. So with this unit in genetics, we are going to be testing whether or not offspring followed our prediction based on a Punnett square or a dihybrid cross. So in order to be able to test this, we are going to be using chi-squares. And to start with, I want to just kind of remind you what a null hypothesis is when we do chi-squares. So typically we think of null hypotheses when we're doing these chi-squares as saying that there's no difference between group A and group B, that there's no difference between what we are expecting to see and what we actually get. And so in genetics, the way that it works is we are now going to be using predictions from a Punnett square dihybrid cross to determine the expected values. So our null hypothesis is that there will be no difference between the offspring produced and what is predicted from a Punnett square or a dihybrid cross. And when we test these predictions, we're always going to be looking at phenotypes. Now, sometimes the phenotypes, we can't really determine what a phenotype is, especially in the case of the dominant trait. So we'll have to do what's called a test cross every now and then to figure out what a dominant phenotype is. And that's going to be because we have no way of determining if an individual who's dominant in their phenotype is AA homozygous dominant or a little a heterozygous. And so with a test cross, what you do is you take a dominant and unknown dominant and cross it with a recessive. If any of the babies end up having recessive phenotypes, then you know that the unknown parent must have been heterozygous. Now, when we're doing these test crosses, or when we're doing this testing, it'll be very helpful for you to get quick at dihybrid crosses. And there's a couple of dihybrid crosses that if you know the parents' genotypes are a certain way, then you can quickly produce or quickly predict what the offspring are going to be. And so these are just a couple of dihybrid crosses I think that are going to be very helpful to you. The first is if both parents are heterozygous for both traits. If both parents are heterozygous for both traits, then the babies are always going to follow a 9:3:1 ratio for their phenotype. The second one that I think is very helpful for you to remember is if you have a heterozygote parent, so heterozygote for both traits, and a completely homozygous recessive parent for the other one. When you have a cross like this, their ratio is always going to be a 1:1:1:1:1:1 ratio. Now, you don't have to memorize these. You can always just do the dihybrid cross. This would just save you some time, and time is something that we don't have a lot of on this exam. So if you can memorize that this cross produces this ratio, and this cross produces this ratio, it's going to save you some time. I hope this video was helpful. Thanks for watching. Bye.